Hi, how's it going, AP Bio kids? Okay, so I've gotten a lot of you reaching out. By a lot of you, since there's three of you, I've gotten you guys reaching out needing help on labs. So I'm gonna make some tutorial videos that'll hopefully walk you through some labs and simplify it so that you can complete them in time before the end of the quarter. So I'm first gonna start with the Population Genetics Lab 1.12, and this video will be for exercise A only. I'll make a separate video for exercise B. So for exercise A, we're looking at Hardy-Weinberg and the principle of Hardy-Weinberg and how to calculate the frequency of alleles in a population and genotypes in a population. And remember, Hardy-Weinberg is like perfect world scenario. Unfortunately, that doesn't happen in the real world because organisms are always evolving, always ever changing. So this is basically just a prediction that you can make as a baseline. It tells you that in the directions and through the walkthrough of the mathematical part of Hardy-Weinberg of why that's important. So I'm going to basically use this time to go over the lab and what is expected of you from the lab so that we can get on the same page here. So if we go and move to the exercise A, Hardy-Weinberg equilibrium, we're looking at the data table that looks like this. Let me pull it up for you. Okay, so we're looking at this part of our document, and I'm going to walk you through and explain what each of these numbers means in the first line. I actually think that I have this open so that I can edit it with you. So we'll go over that in just a second. Okay, so what we're going to be doing is making sure that we can fill out this table appropriately based on the numbers that are asked of you. So looking at the procedure, it tells us a little bit of background. We're working with a population of island jackrabbits, and we're looking at the length of their ears that's controlled by a single gene. So that's going to be the alleles that we're looking at and the genotypes that we're looking at. For the materials for this, it asks you for black and white beans or other objects. So I don't personally have black and white beans just hanging out at my house. So I just looked around my office and I found that I have two things, okay? I have these little black paper clip spiral things and pink. And as long as they're different colors, you can use whatever you have. You could even use pennies versus quarters. As long as there's two distinguishing things, okay? But you want them to be relatively the same size and shape because you're going to be dragging them out of a bag at random. And the point is that it's random. You don't want to manipulate it by being able to determine difference, okay? All right, so just as a preface, if you need to substitute things for the materials lists at times, that's totally okay. The point is just for you to get the correct data. Okay, and obviously you should know that the lab document that you're supposed to submit to the Dropbox has nothing to do with the data tables. The data tables are just helping you get to the analysis part, which is the part of the document that you are turning in for AP Bio. Okay, so we're going to be looking at this part right here. It says, the black beans represent the long hair allele and the white beans represent the short hair allele. So again, I'm working with black and pink, so that's how I'm going to refer to them. The black allele is going to be my long ear allele. So what I did is I made myself a little post-it note for long ears with a capital A for dominant, and I put my black paper clips near it just so that I can stay organized. This is just me because I'm a super visual person. You don't have to take these steps. Then I made a post-it note for short ears with the lowercase a for the allele for the short ears, and I put my pink paper clips under that post-it. Now it says in the directions, set up your initial population by placing 60 black beans and 40 white beans in a bowl. Holy moly, I don't have 60 and 48, but I do have 6 and 10, and percentage-wise, that's the same, which is what we're dealing with. So I took 6 of the black ones and 4 of the pink ones to model the same type of population, and they already recorded them for you in the data table here. So here's our 0.6, here's our 0.4. And I'm using this weird editor because I can write in it. So just bear with me, but this is the same thing that you're looking at, okay? I'm gonna try and minimize this. As you can see, I already tried to make videos for other labs, but haven't necessarily been able to make it work. Okay, I think I might just have to leave it like this. All right, so here's our 0.6 that represents the 60% of the black beans or my black paper clips in my bag. And then I have 4 pink paper clips right here that I'm going to also put in my bag to represent my 40%, which is also recorded in your table already. Then it says step 3, calculate the predicted genotype frequency using the Hardy-Weinberg equation. Anytime you see predicted, it's talking about Hardy-Weinberg because that's what would happen in a perfect world mathematically. So we're using the math to say this is what should happen under perfect circumstances, okay? So let's go look back at our data table and how they filled this out. I'll explain the math to you as we go along, okay? Let me pull up my calculator and I'll show you guys how that works as well. Okay, so we have the predicted genotypes and number next to the frequency. So here's our homozygous dominant genotype. Okay, so it's asking me what is the math behind the homozygous dominant genotype and how many individuals in the population should come out homozygous dominant versus its frequency. So frequency is really easy because we already have the 0.6 allele frequency for our dominant allele. So all I need to do is take 0.6 and square it, right? Remember for Hardy-Weinberg, we have P plus Q equals 1, P squared plus 2PQ plus Q squared equals 1. So those are the numbers that I'm looking at here. And to give you guys a little bit of a cheat, all right, yeah, that's fine. When we're looking at this predicted homozygous dominant, when you should think of this as P squared. I'm just going to have to do the longhand version of this. So this should be your P squared here, okay? All right, so that's our homozygous dominant. 0.6 squared will give us 0.36. I'll explain where they got the 18 from in a second. All right, so now we do the heterozygous. So capital A and lowercase a. And if you look at your Hardy-Weinberg equation, the variable that represents heterozygous is 2PQ. 2PQ. Volts a little better. Okay, so I'm kind of just putting these here as a note for you, okay? And then our last one is our homozygous recessive, and according to the Hardy-Weinberg equation, that is Q squared. Okay, so there are our little cheat sheets. So what we're going to do is we are going to finish calculating these out. For 2PQ, we multiply 2 times P, which is 0.6, times Q, which is 0.4, and that will give us 0.48. Okay, and then for homozygous recessive, we're going to take Q squared, and that's our lowercase a here. So 0.4 squared is going to be, if you don't know that off the top of your head, 0.16. Okay, put it in your calculator if you don't know. So how do we get these numbers here? Okay, well, if you notice, those numbers add up to 50, and the reason they add up to 50 is because you're putting 100 alleles, or according to the directions, you're supposed to be putting 60 black beans and 40 white beans in a bowl. So that's 100 alleles. If you pair them up, that gives you 50 genotypes. So basically it's saying in a perfect world, how many genotypes will come out of this population? So in order to determine that, all you need to do is take your 0.36 frequency and multiply it times 50, because that's how many genotype possibilities there are in this population. So 0.36 times 50 will give us 18. We do the same for heterozygous and for homozygous recessive. For heterozygous, it's 0.48 times 50. And for homozygous recessive, it's 0.16 times 50, and that gives us 8. So that is our chart filled out based on the math. So that's when you have to remember these equations, or these variables in our equation for Hardy-Weinberg. Now it's asking us to do the observed genotype and look at the number and the frequency, or calculate both of those there. This is where we have to go back to the directions. So it's telling us we're going to create tally marks on a piece of paper for each corresponding genotype. So if I were you, something that I would do is I would take Post-its, because I love Post-its, and on one Post-it, I'm going to put the genotype for homozygous dominant, and I'm going to put tally marks on that Post-it every time I draw out 2 paper clips that represent the long ear alleles. I'm going to do the same thing for heterozygous, big A, little a, on a Post-it note, and then I'm going to put my tallies on that one for when I pull a black and a pink. and another one for homozygous recessive. You get the idea. And then I'm going to tally on that one. So then it tells me to do that. Okay, it's going to say pull out 2 at random, and then you're going to record how many of each genotype you pull. And you're going to have to draw out 50 individuals. So because I cheated and I only did 6 and 4, I'm going to pull out 2. Let's do, my first one is pink, and my second one is black. That represents a heterozygous individual, okay, pink and black. So I'm going to put a tally mark on that Post-it. And now I'm going to throw my 2 paper clips that I just pulled, my 2 that represent heterozygous, put them back in my bag, shake it up, and pull again. And I'm going to do that 50 times, which I know sounds a lot. I got homozygous dominant this time. I have 2 black paper clips, so I'm going to record that tally. And I'm going to keep going, okay? Once I have all 50 tallies, I'm going to add them up and put them in my data table here. So that's where it says I pulled 18. I pulled 18 homozygous dominant, 26 heterozygous, and 6 homozygous recessive. Those should be the first numbers that you enter into your data table. The second thing that you're going to do is then calculate the frequency for the observed genotypes. So in order to do this, you're going to basically divide your number of dominant, let's say homozygous dominant for the first one, by the total number of individuals. So that's really easy. 18 out of 50 should give us 0.36. I have so many calculators open. 18 divided by 50. There's our frequency. That's so easy, right? Then we do the same thing. 26 divided by 50 will give us 0.52. And then we have 6 out of 50 should give us 0.12. So you can see the numbers are fairly close, but not exactly the same. And then we move on to the last 2 columns, which are explained in the procedure for how to calculate the observed allele frequency. There's actually an equation that you follow that they give you on step number 5 for exercise A, where to calculate for P, remember P is representing the dominant allele. We're going to take 2 times the number of homozygous dominant individuals that we pulled. So 18 times 2. We're going to add to that the number of heterozygous that we pulled. So add 26. That gives us 62 divided by 100, which we should know is 0.62. So there's our observed allele frequency. And then we're going to do the same thing for Q. Remember, Q represents our lowercase a. If you need to make a little note at the top for yourself, please do P and Q here. We're going to do the same thing. We're going to do 2 times the amount of homozygous recessive we had, which is 6. We're going to add the number of heterozygous to that. Remember, because heterozygous has both of the alleles, so we have to factor that into consideration because it has half of the alleles for that genotype. And then we're going to divide by 100, which will give us 0.38. And that's how we get that frequency. So then the next important part is that whatever observed allele frequency you get at the end of a parent population, in this case the example, you're going to use that for the next generation. So here I'm going to carry down my observed allele frequency 0.62, and I'm going to put that right here, 0.62. Remember your leading zeros. I'm always so bad at that. I won't even lie. So my 0.62 is going to go there, and then I'm going to put my 0.38 in the next column. And I'm just going to start this whole process over again. I'm going to do my second column on predictions based on the Hardy-Weinberg equation, and then I'm going to do the observed by pulling those paper clips out of the bag that I currently have. Okay, so hopefully that explains it. When you finish your data table, it's telling you you're going to answer questions 1 and 2 in your lab report. So here we are. Here's our lab report. Here's questions 1 and questions 2 that pertain to that activity. All right, follow the next video to see exercise B.